Now we're all ready to go. Okay, so the digestive tract is kind of suspended in the abdomen by these folds of tissue that we call the mesenteries. So, we know that the digestive tract is a long muscular tube, right? So if you think of a garden hose, it's also a tube. And you could jumble up the garden hose and you could hang it from the wall of the garage or from the wall of the outside, right? It would sort of dangle like that. Well, essentially, that is how the digestive tract is supported in the abdomen. It isn't firmly attached to the wall. Instead, it kind of hangs from the back wall. So what we're looking at here is a sagittal section. So the person has been cut left and right. We're looking at it from the side. Here's the liver. We're going to talk much more about that next time. And here's the stomach right here. Large intestine here. This is all small intestine here. And you can see that these structures or organs are kind of hung from something else. So like the stomach hangs down from the liver. The transverse colon or the large intestine, it hangs here from the back wall. And then the small intestine, hangs here by sort of multiple hooks. So the mesenteries are these folds of tissue here that suspend the GI tract in the abdomen. So the reason that this arrangement evolved is as materials pass through the digestive tract has to be able to move. So in order to move, its attachments have to be flexible. So this arrangement means that the areas of the small intestine and stomach and large intestine, they can kind of move around each other as things pass through, as they change their shape. So most of the organs of the GI tract are found sort of suspended in this abdominal cavity in that fashion. So we call this cavity that these things are hanging in the peritoneum. So that word is right here, peritoneum. And so the area in between these digestive system organs, so like all this area here, it's sort of in a dark red in this picture. All of that area is called the peritoneal cavity. Normally, the peritoneal cavity is mostly empty. All it contains is a small amount of slippery fluid. So kind of like we saw with the pericardial sac and the pleural space in the lungs, here's another region that is supposed to be empty, but exists so that things can move separate from one another. So the peritoneum comes up clinically with some regularity because anytime there's a GI tract problem, very often the peritoneum, which is the lining of this cavity, right? So this is the peritoneal cavity, and lining it is the peritoneum. The peritoneum gets inflamed or angry. And when that happens, it generates a lot of pain for the patient. It also triggers reflex responses that make the abdomen very hard to the touch. Normally the abdominal muscles are relaxed unless we're using them. Well, if there's any kind of a peritoneal disease process going on, those abdominal muscles get as tight as they can in an attempt to protect this region from further damage. So like in an appendicitis or a ruptured appendicitis, the classic, exam finding is a rock-hard abdomen, where their abdomen is very, very hard, and that's because the muscles are contracting in response to injury to this peritoneal lining of this peritoneal cavity. So the mesenteries hang the GI tract in the peritoneal cavity. That's sort of the short version. Now, it is important to note that there are a couple of digestive system parts that are not suspended out here, but are instead behind that cavity. Okay, so if this is the cavity in red, well, what are these two things? The pancreas and the first part of the duodenum, which is the first part of the small intestine, they are both what we call retroperitoneal. In other words, they lie behind the peritoneal cavity. And again, clinically, that can matter because like when the pancreas is sick, instead of causing peritoneal problems, because it isn't in the peritoneum, it causes flank pain instead, because it's behind it. Another important set of organs that are behind the peritoneal cavity are the kidneys, which we'll talk more about when we get to that chapter. But they also sit behind the peritoneal cavity. All right, so peritoneum, GI tract, mesenteries, and the peritoneum. Okay. All right, so we talked last time about the esophagus and swallowing. So at the end of that swallowing reflex, that deglutition reflex, the food bolus is deposited into the stomach. So the stomach has various regions, and the anatomy on this slide and the next one are worth knowing. The stomach is usually described as J-shaped. So we have, a kind of J shape like this. Here's the esophagus coming down. You can't see it very well in this picture, but it has come down through the diaphragm, right? And then it has then entered the stomach here. The region where the esophagus meets the stomach, we call that the cardia. Not cardio, but cardia, all right? And that's just because it's near the heart, so it got that name. So that's the cardia. This dome-shaped region of the stomach, above where the esophagus enters, we call that the fundus. Fundus just means dome. Okay, so we're gonna see that there are fundi. That's the plural of fundus. There are fundi in several organs. This is just one. So like the uterus, for example, also has a fundus. So fundus just means dome, and in the stomach, that's this part right here. When you look at an X-ray of the abdomen in a patient who's sitting upright or standing, there is usually air in the fundus. So you can see that dome shape on that X-ray, because on X-ray, air shows up black, and other things, bone shows up white, and like blood shows up somewhere in between there. So we often can see the fundus when we look at X-rays. I don't have one to show you. I wish I did, but it's something to see. All right, so that's fundus. The body of the stomach is this region right here, so it is sort of squarish. And then at the end of the stomach here, we have the pylorus region, which is right here, which is the exit from the stomach, so to speak. All right. And then we have two curvatures. So we have a lesser curvature that goes on the small side, and then we have a greater curvature, which makes up the outside of the J. So greater and lesser, and then the regions of the stomach. All right. The stomach ends where it connects with the duodenum, and the duodenum is the first part of the small intestine. And between the end of the stomach and the duodenum, there's a valve called the pyloric sphincter. So if we now sort of cut into the stomach, right, here's the duodenum, right here, that first part of the small intestine. Here's the pylorus, this region right here, the exit part of the stomach. And then between those two, you can see this sort of lump of tissue. Well, it's a sphincter. It goes all the way around that tube, and it can open and close. And it's critical to controlling the rate at which the stomach empties into the duodenum for the continuing of the digestive processes. All right, so that's the pyloric sphincter right here. The pyloric region, or pyloric antrum, which is right here, is kind of funnel-shaped. We have a large part of the stomach that's going to funnel in to a much smaller exit. So we need to have a funnel. So it's the pylorus that sort of makes up that funnel. All right. Here's the esophagus coming in. Here's the cardia, right, where the esophagus meets the stomach. Here's the fundus again. The body is this region right here. Greater curvature, lesser curvature, many of the same structures. Okay. So if we look on the inside of the stomach, what we're going to see is wrinkles. And we're going to see folds and wrinkles as a theme in the GI tract, like we already talked about, folds of folds of folds. In the stomach, we call those folds rugae, and they are present when the stomach is contracted. All right, so one of the stomach's principal jobs is to store undigested food between meals. We don't have to eat, every hour or even every 2 hours. We can go 3 to 4 hours between meals during the day. And part of the reason for that is because the stomach is expandable. So when the stomach is empty, it is contracted and it's its smallest size, and it has these deep folds on the inside. But as we eat a meal, the muscle of the stomach relaxes, and the stomach gets larger and larger with each bite. When it reaches its sort of maximum size, the rugae are no longer present, they're flat. So, if you were, imagine stretching a thing out, as you stretch it, the wrinkles disappear. So the same thing happens to the rugae in the stomach. So they're present when the stomach is empty, but not when it's relaxed and full. All right, so the stomach has a very muscular organ. So it has 3 layers of smooth muscle. It has an outermost layer, which is called the longitudinal layer here. It has a circular layer, and then it has an extra layer compared to other areas of the GI tract, and we call that the oblique layer. The reason for all this muscle is twofold. One is so it can relax and get bigger, right? But the other reason is the stomach completes the process of mechanical processing. In other words, when we take a bite, the first thing we do is chew, 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 chew. Well, that mixing and crushing process is continued in the stomach. So the stomach contracts and moves and squishes and squeezes what's inside it, all as part of that digestive process. All right? So the reason it's muscular is for those 2 reasons. So like, your cats and dogs, when they eat, they don't chew very much, right? So, how does that food get processed? Well, their stomachs do the chewing for them. They squeeze and bend and twist to basically crush up and mix up anything that's been taken in. All right, so our 3 layers, the anatomy. Okay.
When they have bariatric surgery,
Mm-hmm. Banding?
Yeah. What part? Is that at the top?
Yeah, they, I don't know for sure, but I think that they put a band like around here. Because what they try to do is they try to leave just this part open. Because the idea is if you make the stomach smaller, people will stop eating with less food. Because one of the signals for satiety, in other words, I'm not hungry anymore, is when the stomach gets stretched out, that sends a signal to the brain, okay, enough, don't eat anymore. So if you make the stomach smaller, fewer calories trigger that effect. That's the idea behind banding. But I'm not sure, to tell you the truth, where exactly they put the band. They may even just go all the way around to prevent the stomach from expanding, which would also be effective. But
How did they do the old-fashioned bariatric where they would cut the stomach in half or staple the stomach in half?
There again, they would just reshape it. They would cut part of it off and sew it back up, but that didn't work very well. People had massive complications. All right. Okay, so the stomach is a very rough neighborhood, as we're going to talk about in a little bit, because it's in the stomach that we have a couple of different processes that happen all at once. One we've all heard about, and that's the stomach creates acid. And the reason it does that is principally for 2 reasons. One is to destroy bacteria that might be present in the food. Acid is a very powerful weapon against bacteria. And the other is to start the process of protein digestion. Some of what we eat contains proteins, and proteins, in order to be digested, remember digestion is breaking up into smaller pieces, right? You have to uncoil and unwind the proteins to make that happen. And acid is good at that. That's called denaturing. But, so we have protein digestion and acid present. Well, the cells of our body are made principally of protein, right? So why doesn't the cells of the walls of the stomach digest themselves? And this is a recurrent theme in the GI tract because what we need to do is we need to digest food that we've eaten, but food that we've eaten is made of the same stuff we are. Excuse me, I got a sneeze that's coming on here. So we have to have arrangements so that we can digest what we eat, but we don't digest our own cells, our own organ systems, our own digestive tract. So the stomach is the first really good example of that. So remember when we started last time, we looked at the 3 layers of the GI tract, the mucosa, the submucosa, and the muscularis externa, and then the serosa on the outside. Well, for the stomach, the mucosa, which is this part right here, is very, very thick and complex. And the reason for that thick and complex arrangement is to protect these underlying tissues from the acid and protein digesting enzymes that are present in the lumen, which is up here. So this is where the things we've eaten end up. So that's this region here. So we have, we still have those 3 layers, but the top layer, the mucosa, is sort of complexly arranged, which we're going to talk more about in a minute. All right. So if we zoom in to one region of this mucosa, we can see its patterns. And that is, we have pits and we have glands. All right, so this is the inside surface of the stomach here, right? And diving down from that surface, this opening right here, so this region, that is a gastric pit. And at the end, at the bottom of the pit, so to speak, we have glands that open into the bottom of that pit. All right, so if we look at the organization here, you can see we have a single layer of columnar epithelial cells, right? So here's a layer, and here's a layer. Just like you looked at under the microscope last semester, where you're looking at epithelial cells, here's a classic mucous membrane, where we have a single layer of epithelial cells that sit between the contents of the stomach and then the more internal tissues. All right, so those guys are in white, the epithelial cells. They create a thick mucus, and that mucus is very alkaline. So lining the surface of the stomach here, or the inside surface, we have a thick layer of mucus. Acid doesn't penetrate mucus very well. So that is one barrier to the acid that's present out here. And then the other barrier is the alkalinity. So as acid ions try to penetrate this mucosa, they're neutralized by that alkalinity. So those are 2 defenses against acid. So, and then the third one is cell division, which we'll talk more about in a minute. So a very common clinical problem with the stomach is ulcer disease. And ulcer disease happens either when there is overproduction of acid or there's an underproduction of the acid protectors. So, for example, if you turn down the production of that mucus that I was just talking about, that means that acid can get to the mucosa and start to destroy those cells. If all of the mucosa is eaten away, what lies underneath it is arteries and veins. So it triggers a hemorrhage, a massive bleed, due to an ulceration is just a breakdown in this mucus lining. So, clinically relevant, these acid protection schemes. All right, so those are the mucus epithelial cells, gastric pits. All right, so opening into the bottom of those pits then are glands. And these glands, because this pit is so narrow, it's hard for acid to get down here. It's essentially blocked off all the time by mucus. So these cells can live without as much concern or without as much damage from the acidic environment. So one of the body's tricks to dealing with a harsh environment, like the skin surface, right, is with cell division. If you keep replacing cells with new cells, then no one cell is ever very old. So it's a sort of dynamic turnover. So in the gastric mucosa, cell division occurs at this region right here, at the gastric, at the neck, we call that, the neck of the gastric pit, so right here. So as these cells divide, they're pushed up and they're pushed down. So new cells are constantly being made here, and then they end up at the top where they're destroyed and let go. And then new cells also move down here to replace these cells of the gastric glands. All right, so there's this constant replacement. We're going to see that same thing in the small and large intestine, that because cells are being replaced all the time, they can maintain this barrier even though the contents are very caustic or are very harmful, damaging, acids, enzymes that digest, things like that. All right. So when we get down into the glands, we get to cells that make stuff. A gland, by definition, secretes something, okay? So here we have 3 different cell types, and I do want you to know these 3 cell types in the gastric glands. The first are the parietal cells. The parietal cells do 2 things. One thing they're famous for, another thing they're not, but is important enough to remember. Okay, so parietal cells, they are the principal acid producers in the gastric glands. So they produce hydrochloric acid, and we're going to see a picture of that coming up next. But parietal cells also secrete something called intrinsic factor. And right next to intrinsic factor, you should make a little note, relates to B12. So vitamin B12 is a vitamin that's only found from animal sources. And it is a vitamin. We need a small amount of it in our diet. But it cannot be absorbed without this intrinsic factor. So a very common disease in the past, not so much anymore, was vitamin B12 deficiency because of a loss of intrinsic factor. So what do I want you to know about intrinsic factor? Without it, you can't absorb B12. And without B12, you end up with a short list of significant problems, which we're not going to talk about right now. So intrinsic factor goes with B12. Okay, so that's the parietal cells. Your book has them in different colors. Of course, in real life, they're not different colors, but they're in approximately these locations. The parietal cells are up towards the top, and then the G cells are way down at the bottom. Okay, so those are parietal cells. The pink cells here, so the parietal cells are in orange, pink ones are called chief cells, and their claim to fame is they produce pepsinogen. So we've talked before about anytime you see this O-gen at the end of a protein name, that means that it's an inactive precursor. So pepsinogen is released, it travels up the gland, it's released up here, and acid in the stomach turns pepsinogen into pepsin. And pepsin digests proteins. So pepsin begins that process of protein digestion by turning large protein pieces into smaller ones. All right, so pepsinogen becomes pepsin. And what does that is HCl, hydrochloric acid, right? And then pepsin turns big proteins into small ones. So it breaks proteins up. All right, so, and that is the first place in the digestive tract where protein digestion begins. Because in the mouth, there aren't any enzymes to begin that process of protein digestion. It isn't until we get to the stomach that happens. And the stomach is a good place for that because the acid in the stomach also denatures the proteins, uncoils them so that pepsin can get in there and start breaking it up. All right. So I do want you to know parietal cells, we haven't talked about G cells yet. G cells produce hormones. The most important one they make is gastrin. G is for gastric, G is for gastrin. So G cells produce gastrin. Gastrin is the stomach hormone. It stimulates everything the stomach does. So it stimulates motility, how the stomach moves. It stimulates the secretion of both that mucus as well as the acid and pepsinogen, which becomes pepsin. So gastrin is sort of the control hormone for the stomach. And yeah,
What is that schema file?
big proteins become small.
Okay.
Yeah, that one is particularly bad. Sorry. All right. Okay. Okay. So, how exactly is that stomach acid produced? Because you'd think, well, acid is a simple enough thing. Why is that interesting? Well, in our bodies, hydrochloric acid, present in our cells would be catastrophic, right? Because HCl is very, very acidic, and it would cause all kinds of problems. So the only place we can really have that hydrochloric acid is behind those acid barriers that we were just talking about. So what the parietal cells do is they kind of do this sort of neat trick, and that is they take a carbon dioxide molecule, which there's always plenty of around, right? Add it to a water, they make carbonic acid. This should look familiar because it's exactly what we talked about with the respiratory system. But here, we're going to drive that equation towards bicarbonate, HCO3 and H+, and here's where the neat trick comes along. We're going to send these two ions in different directions. We're going to pump the proton into the lumen of the stomach, and we're going to pump the bicarbonate into the bloodstream. So essentially, we've taken an acid, carbonic acid, we've split it, and we've sent the two pieces in two different directions. So the acid goes into the stomach, the bicarbonate goes into the bloodstream, all from a molecule of CO2. So it's kind of a nifty plan. Now, as the bicarbonate is shuttled out by this protein right here, a chloride comes in, a minus for a minus. We see this in cells a lot, that if you're going to move an ion, oftentimes you're going to move an ion with the same charge along the other way, right, to keep the cell electrically where it should be. Because if all you did was shuttle out a bunch of negative charges, the cell would become more and more positive, and it doesn't like that. So as the bicarbonate goes out, the chloride comes in, and that chloride then diffuses. It follows along behind the H+, and what do we have? We have hydrochloric acid entering the stomach. So we're using that carbonic acid, or carbonic anhydrase, carbonic acid path that we talked about to split the H+ from the bicarbonate. Now what this means is anytime your stomach is secreting acid, which it does in response to food or thoughts of food, smells of food, things like that, it means that your bicarbonate, or the bicarbonate that's coming out of the stomach, is going into the bloodstream. So we call that the bicarbonate tide. So when you eat, the blood that's coming out of your digestive tract is actually very high in bicarbonate because all the H+ that it was made from went into the stomach instead. So we do sometimes see effects from that. So we have the bicarbonate tide here. All right, so why make acid in the first place? Well, this is the short list. One is to denature proteins in order for pepsin to do its job, and pepsin just starts the process of protein digestion, but we have to uncoil those proteins first. So denatures proteins, destroys almost all bacteria, the hydrochloric acid does. So it means that the food we eat is not sterile. Sometimes we think it is, but it isn't. But by the time it leaves the stomach, it's mostly sterile because the hydrochloric acid destroys all that. And then finally, it's the acid, as we see over there on the board, that turns the pepsinogen into pepsin. And then pepsin starts to break down proteins. In the end, we have to turn proteins, long chains of amino acids, into individual amino acids. So going from big proteins to small proteins is a step in the right direction, but it's just the beginning of that process, as we'll see in the rest of this chapter and in the next chapter when we talk about the rest of protein digestion. All right. So parietal cells do this, put the H plus and bicarbonate in 2 different spaces. Okay. So from the stomach, the next step is the small intestine. And the small intestine has 3 parts, 3 regions that are slightly different in their specializations. But in general, the small intestine, one of its specialization is folds of folds of folds, which we introduced a little bit on Monday. And the reason for the folding is all the 2 most important aspects of the digestive tract happen at the surface, right? And that is digestion and absorption. So because that's the case, we want to have as much surface area as possible. So I talked about the wrinkled sheet last time. We see a lot of wrinkles in the small intestine in particular. So pooching out from the walls, we have fixed wrinkles called plica circularis. Unlike the rugae of the stomach, the plica are always present. So whether the intestine is full or empty, the plica circularis are present. And then lining the plica and the wall of the tube is this kind of carpet-like arrangement of villi, right? So here are the villi sticking up. And this whole region with the carpet here, that's the mucosa. Underneath it, the submucosa. And then underneath that, the muscularis externa. So our classic 4 regions of the small intestine. All right. So folds of folds of folds. All right. So the arrangement of the mucosa in the intestine is we have these villi that stick up, and depending on where you're at in the intestine, the villi can be quite long, even macroscopically visible. Like if you look at it with the naked eye, you can see these sort of finger-like things that stick out. So we have villi that stick up, and then in between the villi, you can see these little pits or holes or crevices. We call those crypts, C-R-Y-P-T-S. Like ancient Egyptians have crypts. Well, the intestine has crypts too, and similar to the gastric glands, down here in these crypts, that's where cell division occurs. So what we have is cells divide here and get pushed as new cells come along behind them. They get pushed up and out, and by the time they get to the top of the villus, they're shed, they're released, they're destroyed because new cells are coming along behind all the time. So it's like a conveyor belt. Here's where the cells are made, and here's where they're released into the small intestine. Now, why would we do that? Because what's going past this region in here, so what's going through the small intestine, is particularly toxic to cells. You're talking about digestive enzymes that are going to pull proteins apart, enzymes that are going to destroy carbohydrates, digest fats. You've got acid coming into the duodenum. You've got alkaline solution coming from the pancreas. It's a terrible place for a cell to live. So the way the body handles that is by not having any one cell live there very long, right? So the cell division rate in the intestine is one of the highest in the body. So we have, hundreds of millions of new cells are being created in these intestinal crypts every day and then released at the villi in order to keep this mucous membrane that lines these villi fresh and new and intact. So like in chemotherapy, chemotherapy's target is rapidly dividing cells, right? Because cancer cells are usually in that category. So we give somebody chemotherapy because we're trying to kill their tumor. Almost always, the intestinal epithelium is affected as well, because even though it isn't a tumor, it is rapidly dividing cells. And it has to work like that in order for this mucous membrane to stay intact. So one of the most common side effects of chemotherapy are intestinal problems. So namely, terrible diarrhea, malabsorption, where you don't absorb the food that you eat, so you get a lot of weight loss, You can even get blood in the stools if this mucous membrane starts to break down. So it's one of the limiting factors. Why can't we just give people so much chemo that the tumor dies instantly? Because you kill the intestinal epithelium and therefore the patient. So one of those spots. Another spot of rapid cell division that we've already talked about is the bone marrow. So the bone marrow is constantly producing new cells. So is the GI tract. And in the small intestine, it's down here at these crypts or pits. All right. So underneath the mucosa, we've got the submucosa with artery, vein, and lymphatic. And then we have our two layers of muscularis, or smooth muscle, that are going to cause peristalsis, like we talked about last time, and segmentation, or that mixing action. All right, so specializations of the intestine. All right, so now if we take one of these villi and we zoom in on it, now we can really start to see the structure that makes the function, right? Okay, so this is a single villus. If you look along the border here, this is our epithelial layer, right? One cell thick. These are columnar, simple columnar epithelium that line the intestine. And if you look carefully, which you can't really see in this picture, but there's a fuzzy edge, right, along the outside of that border. And what you're looking at there is the columnar epithelial cells that make up that border. If you were to zoom in on them, they look like this. So they look a little bit like Bart Simpson, right? So they have a flat piece, that's the inside. So that's this side of the cell. And then on the outside of the cell, which is pointing towards the lumen, we have, these are called microvilli. So the walls have villi that stick up, and the cells also have villi at the top. And it's for the same reason, it's surface area expansion. By having a folded roof or a folded top to this cell, you'd have a nucleus like that, we get more surface area here at the top. So when we look at the epithelial border under the microscope, unless you have a perfect specimen and a perfect microscope, you can't see the individual microvilli. Instead, it just looks like this. It's sort of rough, and so we call it, looks like a brush. So one of the common words that we use to describe the intestinal epithelium is we call it the brush border. So the brush border are the microvilli of the columnar epithelial cells that make a single layer that cover these villi. And then the villi cover the plica circularis and the inside of all of the small intestine's tubes. All right, so the brush border, that's these guys. Scattered throughout that brush border, like here and here and here in your book, it's they're in purple. Those are mucus cells. So just like we saw in the respiratory tract, how we had one type of cell, those ciliated cells, and then we had mucus cells every so often. Same arrangement, we have mucus cells every so often here, and that mucus helps to protect the these epithelial cells from the damaging contents that are out here in the small intestine. All right. So everything that enters the bloodstream has to pass through these cells, not around them, but through them. So we have two important features. What do these intestinal epithelial cells do? One is absorb. So if we have a nutrient molecule that's up here in the small intestine, to bring it into the blood, we're going to go through this cell. So these cells create an active membrane that selectively brings things into the bloodstream or blocks things from coming in. So stuff has to go through these cells. And then the other important feature is at this brush border area, these cells have enzymes that stick out. And those enzymes that stick out complete the process of digestion. So like pepsin, for example, we talked about, turns big proteins into smaller proteins. There are some other enzymes which take those pieces and make them smaller and smaller and smaller. But the enzymes that turn proteins into single amino acids are found at that brush border. They're found on those microvilli that stick up and out. And we're going to talk more about the details of that digestion piece when we get to the next chapter in metabolism and energetics. All right, so mucus cells, epithelial cells, brush border. So in the center of this villus, we have this kind of complex-looking arrangement of vessels, right? Remember, one of the roles of the GI tract is to absorb nutrients, to get nutrients into the bloodstream so that they can be distributed to all the cells of the body. Well, that's what this capillary network is here for. So as substances pass through the epithelial lining here, they are then they enter into the bloodstream, go off to the liver, and then circulate around the body. And then right in the center here, we have this big green thing. The lymphatic system in the intestine is somewhat modified compared to what you see in other places. We have this single large sort of tube running through the center of each villus that we call a lacteal. Now, lactose is milk sugar, right? So lacteal must have something to do with milk, and it does. When we eat a meal that has fat in it, the fat that's absorbed is too big to enter the bloodstream. So instead, it enters the lacteal system. The lacteals all combine and eventually join the thoracic duct that then goes up the thorax and into the subclavian. So why these are called lacteals is that fat that's present in there is very white. So the lymph that comes from the small intestine during the digestion and absorption of a fatty meal looks like milk. It looks milk-like. It's white and it's sort of slippery like oil or a fat would be. So we call that single large lymphatic down the center the lacteals. All right, and we'll talk more about how the fats get in there again in the next chapter. This chapter and the one after it sort of dovetail together. All right, so what are the parts of the small intestine? Okay, so here would be the stomach that's been removed, right? And the first part, which is sort of S-shaped, that's here, that's the duodenum. The duodenum becomes the jejunum, which is here in purple. So this whole region is all the jejunum. And then the jejunum becomes the ileum, which then makes up the rest of the small intestine. That's this guy here. It's in like orange. And then the ileum ends when the small intestine ends, where it joins right here at the cecum, where it joins the large intestine. Now, these three regions are not just, convenient names. There are significant differences between the three because of what they have to accomplish. So, like the first part of the small intestine, which is the duodenum, some people call it duodenum, but that isn't how I learned to say it. So, I say duodenum. Duodenum is also fine. Its specializations is it's short. It's only about 6 inches long. The walls of the duodenum are relatively smooth. We were just talking about how the intestine is folds of folds of folds. Well, the exception to that is the duodenum. There are very few villi. There are few plica circularis. And it's because the duodenum's job isn't really to absorb, it's to receive. And that is, it receives the contents of the stomach through that pyloric sphincter. And, it's going to process that content. So, like, what it gets is very acidic from the stomach. So the pancreas neutralizes that acid, and that occurs in the duodenum. So the duodenum is an important region because it's the control center for digestion, as we'll see more next time. But in terms of its anatomy, I want you to know it's relatively short and it has smooth walls as opposed to wrinkly walls like all the rest. All right, so duodenum. The jejunum, its specializations are kind of the opposite of the duodenum. The jejunum is all about absorption. So it has very many folds, folds on folds on folds. Deep plica circularis, very long villi. So it evolved to maximize absorption at that brush border and digestion at that brush border. So the more border there is, the more those two things can happen. And the more folds there are, the more border there is. So it sort of all fits together. So that's this region. And then the last part, the ileum, is kind of in between. It has plica and villi, but they're sort of short. The ileum is the longest region, even though it isn't the most efficient region. So like the jejunum does most of the work of the small intestine. The duodenum just arranges the work. The jejunum does the work, and that is absorption and digestion. And the ileum does whatever is left to do. So it's the longest region, but in some ways, it for every foot, it doesn't do as much as the jejunum. So when we look at these three regions, I'm going to turn this other light off too so you can see it better, because there's our picture. Okay, so if we cut into the duodenum, which is here, you can see how the walls are nice and smooth and sort of shiny, right? And that's because it doesn't, and the plica circularis are very small, and that's because there aren't very many villi there. But then, if we go just a foot down the intestine, and we look at the jejunum, you can see lots of folds, right? And you can't see it in this picture, but it would be like a carpet, like velvet almost, with all of the villi that are present on all those folds. And then down here, if we looked at a piece of ileum and we cut it open, it's sort of in between. It has plica, and it is sort of velvety, because there are villi there, but it isn't smooth and shiny like the duodenum. It's sort of, in between. So all the parts of the small intestine essentially do the same thing, and that's absorption and digestion at the brush border. But as you go along, there are different specializations, so to speak. All right. So let's end with a few questions as we got a little time. Okay, so which of these enhance the absorptive effectiveness of the small intestine? In other words, what makes it work better? All right. And hopefully you'll get the question. I got an answer, so you must have got the question. All right, did everybody answered? All right, good. Yes, E, all of the above. And please, 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 with all of the aboves, be sure you read each answer, because don't just assume that it's correct. Okay, so yes, villi expand surface area, plica circularis are those folds. They don't disappear, so they're always present. Microvilli on the brush border, like we talked about, and then the intestine is always squeezing itself around what's inside it. So that too puts what's to be digested and absorbed right next to where that is going to happen. All right. The double layer of membrane that supports the viscera in the peritoneal cavity are called which of those things? All right, jump in there. All right, so we got two answers, A and D. So the best answer there is D. Mesenteries are sort of two folds of serosa that hang off the back wall and suspend everything in the peritoneal cavity. All right, we're out of time today, folks, so enjoy your weekends, and I will see you on